Many of us are familiar with the scientific breakthroughs within the industry, and one of them is CRISPR gene editing. CRISPR gene editing holds a whole new promise to therapeutic applications, such as modifying the genes of certain CAR T cells so that they can attack cancer, and also modifying other mutations to ensure that cancer is killed with once and for all. In order to better understand CRISPR gene editing, it's important to know that there are a couple of components. One of them is the guide RNA, the other is the target DNA sequence, and the final one is the Cas9 protein. The guide RNA usually matches about the DNA sequence site. This means that if you want to repair a specific sequence, the RNA would release a couple of strands in order to match and guide the Cas9 to the specific site. The Cas9 is basically a micro scissors, which will then be able to cut the sequence. And once we're able to cut the sequence, we can introduce whatever repair we want and produce the desirable trait. One example of mocking CRISPR gene editing is with E. coli. Now, what scientists have been able to do is modify the genes of E. coli so that they produce a resistance against antibacteria. For example, in E. coli, one of the main changes we make is with the chemical compound adenine, or in the entire 4.5 billion letters of the genome of E. coli, that is just an A. And we replace it with the C, which is known as cytosine. Now, by repairing the specific A to C complication, we're then able to produce the amino acid lysine instead of the amino acid threonine. Eventually, this will then produce an antibiotic resistance. By using and inhibiting the specific sequence change, we're then able to develop the antibiotic resistance against the common antibacteria known as streptomycin. Now, what we'll be able to do during the experiment is look at the bacteria population in the specific given medium. Eventually, once we add a couple of droplets or a specific quantity of the Cas9 and guide RNA, we'll notice visible qualitative observations such as color changes and white little mold pieces on top of the bacteria. Then we'll know that CRISPR has done its job, and once we test it against the antibacteria or antibiotics, we'll be able to know that CRISPR holds a whole new promise for decades to come. This will mock many trials of modifying CAR T cells so that they can fight against cancer and also many other mutations in order to help many diseases that kill billions.